Across our planet, life is wrapped in countless bacterial cell walls. These walls are tiny shields built from sugars and short protein bridges. To a bacterium, this mesh is both armor, skeleton, and outer boundary. It holds shape while water presses steadily from every surrounding side. Yet every fortress carries a weakness hidden in its repeating chemical pattern. The weakness is a bond, copied millions of times along sugar chains. Break enough of those bonds, and the wall can no longer hold. Long before laboratories noticed, nature had already found the exact molecular key. In the clear tears of animals, a quiet guardian was working. In saliva, mucus, and milk, the same defense waited patiently nearby. In the white of an egg, it guarded a living possibility. Lysozyme was there, small, ancient, protective, and almost impossible to see. The 19th century brought lenses, stains, incubators, and a startling microbial world. Bacteriologists began to see disease as swarms of living cells. They also noticed that bodies did not stand defenseless against microbial invasion. Blood serum could damage some microbes even outside the living body. Animal secretions sometimes checked growth in ways no one yet understood. These were only clues, scattered like footprints after rain on stone. The actual molecule remained unnamed, folded inside the most ordinary protective fluids. Its discovery came through accident, patience, and one sharply observant physician. In 1921, Alexander Fleming worked at St. Mary's Hospital in busy London. He studied bacteria in a crowded room of plates, glassware, and notes. One culture held a yellow microbe, then called Micrococcus, lysodeikticus by bacteriologists. A drop from Fleming's own nose touched the waiting bacterial lawn. Around the drop, the yellow growth faded into a clear, empty space. Fleming watched a simple human secretion dissolve a living bacterial colony. This was not penicillin, which would arrive several famous years later. It was another sign that nature carried its own protective chemistry. Fleming searched for the substance in tears, saliva, sputum, and egg white. He found it widely, especially in the protective white of hen eggs. He named the agent lysozyme, an enzyme that could lyse cells. The name carried both action and mystery in a single memorable word. Fleming published the work in 1922, describing the clear zones with care. The finding was modest and easily overshadowed by the coming antibiotic age. But lysozyme kept returning to science because it was dependable and visible. Egg white offered researchers a rich, accessible, and familiar source material. During the 1930s, lysozyme was purified and crystallized for much closer study. Its crystals let scientists treat an enzyme like a tiny mineral clue. Decades later, X-ray crystallography revealed its folded three-dimensional form in detail. David Phillips and colleagues mapped lysozyme in the 1960s. It became one of the first enzyme structures ever solved. For biology, this was like seeing a lock from inside. The map showed a groove, shaped to cradle a bacterial wall chain. Along that groove, certain amino acids sat in careful positions. They did not smash the wall, they guided a tiny reaction. A water molecule was invited into exactly the right place. One bond in the wall chain was loosened and split. Then another chain could fail, and the mesh began opening. In a susceptible bacterium, pressure from within finished the work. The cell lost its boundary, and its contents blurred outward. Lysozyme itself remained unchanged, ready to help again and again. Its power came from precision, not force or corrosive strength. In our bodies, this precision works wherever soft tissues meet the world. Tears sweep across the eye, carrying lysozyme over exposed living surfaces. Saliva bathes the mouth, where meals, microbes, and breath constantly meet. Mucus lines passages where each breath brings the outside world inward. Milk carries nourishment and also quiet protection for the young mammal. Egg white surrounds the yolk with water, proteins, and antimicrobial defense. In cheese making, lysozyme helps protect certain cheeses during slow aging. In wine, it can help guide fermentation away from unwanted bacterial turns. In some foods, it supports preservation with a lighter chemical footprint. In laboratories, lysozyme helps open bacterial cells without dramatic force. That simple opening has helped reveal proteins, genes, and inner machinery. Its story also shaped how scientists imagined natural antimicrobial defense. Fleming's clear zone became a window into innate immunity itself. The egg, the tear, and the microbial wall were suddenly connected. Industrial interest grew because lysozyme can work under mild conditions. It could serve processes seeking cleanliness with less dependence on harsh chemistry. The deeper lesson remains older than laboratories, factories, and human plans. In a greener future, enzymes like lysozyme may replace force with finesse. Enzymes Bio exists because good science should be simple to source. We're enthusiasts who put the world's enzymes in one place for startups, labs, and B2B buyers. Kinder chemistry, here for generations to come. Find us at enzymes.bio.